when I first encountered molecular genetics, I remember being in a lecture. This was at Caltech here in California, Southern California. I was in a lecture as a graduate student where the professor was explaining how cells have an ingenious mechanism for controlling the level to which a gene is turned on. So cells can turn genes off or they can turn them on, and they want to turn them on. So I'll speak in teleological terms. A cell wants to turn a gene on if it needs more of the product that that gene makes. Okay, and that's how an engineer would think about this. You want the machine to do this. You want your computer to do this. And when I saw the mechanism, this is at a molecular scale that cells have in order to sense whether they need more of the product of this gene or not, and then control by proteins binding to DNA and RNA, control how much of the product is made, as if it's got it's a regulatory circuit of the kind that I had studied as an engineer. And there's something called control systems theory. I'd studied that as an engineering student, and I thought this is a control system, but it's a control system that's far more remarkable than anything I ever saw before as an engineer, because it's single molecules implementing this control system. And that's what got me thinking, first of all, I do not believe that this just happened by chance. Someone who has the mind of an engineer, but more brilliant than any human engineer, did this. But I also started thinking, how would you go about proving this? Proving that this can't just happen by accident, by a Darwinian process. And that led me down the line of thinking, well, what could you do with single genes in a laboratory that would shed light on this? And this was I first started thinking along these lines in the 1980s, and this was about the time that some very powerful techniques for manipulating DNA were becoming available. So the 1980s was the big boom of biotech, and you had these companies springing up, and it was, cloning and recombinant DNA technology. And I was following all this very closely because I was in a grad school where I was in biotechnology. But I started to become more interested in using these technologies to do an experiment rather than to start a biotech company. And so I went the direction of molecular biology. That's what landed me in Cambridge. But the experiment basically was, can I take a gene that has the sequence instructions for cells to make a protein that does something, and can I mess it up millions of different ways and put these messed up genes in bacterial cells and let them tell me whether the thing still works or not? Because if I can let them do the work of telling me whether it works or not, I can get numbers from an experiment that would allow me to come up with a probability that something that's entirely messed up would or would not work. So that's what got me into proteins. I wasn't a protein scientist who then stumbled upon the question of evolution. It was the other way around. I was fascinated with testing evolution, and that's what led me to study proteins.